Dragana is the recipient of the 2014 Klingenstein-Simons Fellowship Award in the Neurosciences. Her work is about how the brain switches between waking and sleep, since all of us are sleep and are awake. It's important to understand how this operates and why we need sleep anyway. So she's studying three main lines: how our brains fall asleep and wake up, and looking at sleep centers, not in humans but in flies, which makes it a little more amenable to research, and tell us about some of the intricacies of this work. So thank you.
So first of all, thank you so much for inviting me to be here. This is really an incredible honor to be included in this group of amazing scientists and speakers, especially at a meeting that has such an important idea at its heart. And I really want to thank all the women that came before me. Do I need the microphone or did I have that?
It's on now.
Oh, okay. So I can move around. That's better. Yeah, that's what I'm saying. I really want to thank all the women that came before me that fought the hard battles and really paved the way for me to be here, and I think I have it pretty easy. But I should also say that I had really good male mentors, which made all the difference. My PhD advisor, Ken Irvine, and my postdoctoral advisor, Mike Young, have really always treated me as an equal, which I think had a lot to do with where I am today. And I should say that the chair of my department, Mike Greenberg, is really dedicated to diversity and equality. I think he's really passionate about this, and I really appreciate that. So what I'm going to tell you about today is a little bit, just to give you a flavor, because I don't have really much time, but I just want to give you kind of a flavor of the questions that we're asking in my lab. So we're primarily interested in sleep, and I think it's fair to say that among basic animal behaviors, this is the most mysterious one, the most bizarre one. It's really not obvious why it is that we would do this to ourselves every day. Why it is that you would put yourself in danger and put yourself in the state that seemingly doesn't allow you to do anything productive. The questions of what sleep is, how and why we do it, have really captured our collective imagination since the dawn of our civilization. So what I'm showing you here is a 4th century BC Etruscan statue called Sleep and his half-brother Death. So you see these two winged creatures, Sleep and Death, carrying this limp body. And really, for all practical purposes, you can't tell if this person is dead or if he's just sleeping. And a little bit more modern, but along the same lines, is this quote from Shakespeare's Macbeth, in which he refers to sleep as the death of each day's life, sore labor's bath, balm of hurt minds, great nature's second course, chief nourisher in life's feast. What the ancient Etruscans and Shakespeare lacked was the ability to peek inside the brain of a sleeping person the way that we can today. And if we do that, we see that a sleeping brain is very different than a dead brain, but of course is very different than an alive brain. And so this is what we want to understand. We want to understand this process that transforms us so profoundly every single night, that really robs us of everything that makes us truly alive, that makes us who we are, for many hours at a time, but in a quickly reversible manner. So how do we do that? Where do we start? So we've known for a very long time that there's a strong genetic component to sleep regulation. So for instance, we know that many sleep disorders tend to run in families, and we think of the EEG pattern during sleep as being one of the most heritable human traits. So the idea is that if you can find sleep genes and characterize their function, perhaps you can understand something on a deep molecular and mechanistic level about this behavior. Of course, to do that, you need a really good model system. And our model system is the fruit fly. And this is because the fly has really been instrumental for us, for our understanding of biological processes from development to behavior. So I want to tell you, for instance, that many of our behaviors are driven by our circadian clock, which is our endogenous oscillator that keeps us in sync with our cycling environment. So we're exposed to light-dark cycles every day. Here is what the population of Manhattan looks like during the day and during the night when everybody's off sleeping. So sleep is just one obvious output of the clock, and there's many more. And the reason I'm telling you this is because
it was shown the cogs of the clock were first identified, and the regulatory logic of the clock were first worked out in the fly. So here is the Drosophila clock, here is the mammalian clock. The details are really not important, but what's important is that these are largely very similar. Some details are different, but the logic is there. Okay, so the fly has really taught us a lot about biology. And we've known for a very long time that flies have periods of activity and periods of quiescence. But the big question was, when flies are quiescent, are they really sleeping? Okay, does this resemble our sleep? And almost a decade and a half ago was shown that this was really the case. So this is how we measure sleep in the fly. We put them into these glass tubes. This is funny, here it's nice, and then here you can barely see it. Okay, so we put flies into these tubes. There's a lot of food on one end. There's an infrared light beam which bisects the tube. Flies can see that, but each time they walk through, they break the beam, and then the computer records this as movement. And the data are then plotted on an actogram shown here. So each horizontal line here is 1 day of data collection, and each vertical line is 5 minutes of pooled beam crossing data. Is that better? Okay, great. That's better. Okay, thank you. So the experiments are done in 12-hour light, 12-hour dark cycles, and what you can see is that flies have consolidated rest and activity periods. So they can be active for long periods of time, and they can be quiescent for long periods of time. Of course, you can say, well, maybe when the fly is at this state, it's just not moving. It doesn't mean that it's sleeping. Maybe it's just sitting in one end of the tube and not crossing the beam. But what's been shown is that if flies are inactive for 5 minutes or longer, this is associated with an elevated arousal threshold, meaning that you need a stronger stimulus in order to get them to react, which of course is the same thing that happens to us when we're sleeping. So we use 5 minutes of inactivity to define a sleep episode in the fly. And this altered arousal threshold suggests that their brain is at an altered state, and more directly, if we look at the brain electrical activity changes, we see that they differ between waking and sleep states. Fly sleep is under both circadian and homeostatic regulation. So the circadian clock, to which I already alluded, regulates the phasing of sleep. Flies are diurnal animals, just like we are. They're active during the day, sleep at night, which is opposite to many rodents. And then there's also a homeostatic component, which ensures that animals get enough sleep. So if you sleep deprive them, if you cause them to walk around the whole night during the time that they would normally be active, then they sleep the next day during the time, you deprive them during the time that they would normally be sleeping, and then they're sleeping during the time that they would normally be active. So this is rebound sleep, and this is what we all do. So flies need their sleep just like we do. And so all the criteria that people have established to ask whether a period of quiescence really resembles sleep, flies have passed. But of course, the important question is, are we going to be able to use the knowledge that we gain from the fly to understand something about our own sleep? And we are having accumulation of evidence that suggests that this will be the case. So first, if we look at pharmacology, we see that many substances that regulate our sleep do so in the fly as well, and this suggests that the underlying mechanisms of regulation will be conserved. And then more directly, if we look at the genetics, we see that the factors that are known to regulate sleep are conserved between mammals and flies.
I'm sorry, you're giving coffee to the flies?
You can give them caffeine, yeah, and many other substances that regulate our sleep. So some details may be different, but you know, the logic is probably the same. So there are two things that are worth pointing out here, and one is that this is really not the comprehensive list of sleep factors, but the list of factors that we know today is not that much longer than this. So there's really not that many factors that we know. And the other is that many of these that we think of as sleep factors are either expressed broadly or act broadly in the nervous system. So it's often very difficult to tease apart their specific role in the regulation of sleep. So for instance, GABA is the main inhibitory neurotransmitter in our nervous system and in the nervous system of the fly. When you take Ambien, which is perhaps the most widely prescribed sleep medication, this acts to potentiate GABA signaling, and it's great, it can put you to sleep. But there are many side effects associated with taking Ambien because of these non-specific effects. You're kind of dampening the entire brain activity. So it's kind of a sledgehammer approach. And there's really a need for finding and characterizing more specific, more dedicated sleep factors. So when I was a postdoc in Mike Young's lab at Rockefeller, I decided to to take an unbiased approach to look for new sleep factors. So I really didn't want to look under a lamppost. I wanted to keep a completely open mind. And the approach that I took was basically to turn genes off or dial them down one by one, specifically in the nervous system. So flies have different cell types, just like we do, and we have really good technology to target gene expression in any tissue that we want using this GAL4 UAS system. So we have these factors called GAL4, which can be expressed in any tissue of interest. In this case, we're using one that's expressed in the nervous system. And we're using it to express RNAi molecules, which can basically dial down or turn off gene expression. So as I said, we're turning genes off, we're dialing them down one by one in the nervous system. So the advantage of this approach is, first of all, behavioral mutants are very difficult to map. So if you just bombarded DNA with mutagens, got an interesting phenotype, and then tried to go and map where that came from, this is really difficult for behavioral phenotypes. And the other, so if you take this approach, you know already what you're targeting, right? And we have libraries of RNAs that are targeting basically the entire Drosophila genome. And the other reason is that I thought maybe by taking this approach, we can uncover whole new, interesting, and unexpected classes of genes. You can imagine that there could be factors out there that are needed for the animal to develop, okay? And you could never take the function of such genes broadly, because this would be lethal. But if you restrict RNAi in time or space, you can bypass this early lethality. So this is the approach that I took. I screened through about 20,000 animals, almost 4,000 individual genes. And we're now left with a list of 20 new sleep factors that we're studying in my lab. So I'm going to show you some of the phenotypes that I got from this screen. What happened? I'm not sure what you did, but I can't go. I can't advance my slide. No, but this. All right, well, thank you. Okay, so here are the control animals which are active during the day, sleep at night. And in contrast to that, we get these nocturnal animals which are now sleeping during the day and are active at night. So they behave like many lab rodents. We get these sleepy heads. Now they're sleeping a lot, both during the day and during the night. But what's important is that they still wake up at this transition between light and dark, or they're active at this transition, so they respond to environmental photocycles. They're not paralyzed, and this is really important to us. Very different than that, we get these flies that have trouble sleeping. They pretty much don't sleep at all, and there's a very steep price attached to this kind of lifestyle, so their life is very much reduced, and the hope is that by understanding what goes wrong in these animals, perhaps you can understand really what the physiological basis for sleep need is. And we can get more subtle phenotypes, which I'm really excited about. So for instance, these guys have trouble falling asleep. So in the first few hours when the lights are turned off, these controls go asleep quickly. But these guys stay up for a few hours. You see that this peak is extended. Once they go asleep, they stay asleep. Okay, so this is a sleep initiation problem. These guys, on the other hand, have no trouble falling asleep. They fall asleep just fine. So you see in the first few hours, there's lack of activity, but then they wake up and they have trouble staying asleep, so they wake up and they continue waking up. Okay, so this is a sleep maintenance deficit. And this is really exciting for us because it tells us that we can genetically separate initiation from maintenance of sleep. And again, with potential sleep therapies in mind, we think this is important. And it also allows us to ask all kinds of interesting questions. So for instance, for me, it's interesting to think about this notion of falling asleep. We all say fall asleep, I say fall asleep, but I think that this terminology may not be really correct. In a sense, it may be more accurate to say that we walk into sleep, or at least we walk up to a precipice from which we can fall into sleep. And what I mean by that is that when you're going to sleep, there are certain rituals involved in this. You go and you look for a secluded spot, you turn off the light, close your eyes, adopt a specific posture. So you kind of have to induce this transition. And by having these sort of nuanced phenotypes, we can actually ask questions like this. Okay, so we're studying these factors in my lab now, and I'm just going to tell you just a few words about one of the factors that we were really interested in, and that's this factor called cyclin A. So Here are the parental controls, which have the two components of this bipartite system, but they're essentially like wild type, they're controls. And these are the animals in which cyclin A is knocked out from the nervous system. And these animals sleep less. So this is daily sleep plotted here. You can see that they're active on average. They're much more active than the controls. So why this factor? Why was this interesting to me? Well, cyclin A is a core cell cycle factor. So we canonically think of it as being needed to regulate cell division. It acts in proliferating cells to control division, right? So during development. And this really fits this definition of a factor that I had in mind, because if you had a non-mutation in cyclin A, this would be lethal. But because we restricted RNAi to the nervous system, we can uncover the secondary exciting function for this factor as a sleep factor. And there's also a lot of knowledge about this factor from the cell cycle studies. So we don't expect everything to be exactly the same in cell cycle and in neurons, because I should point out again, these cells are not proliferating. These are post-mitotic cells, neurons, right, in which it acts to regulate sleep. But we expect at least part of the framework to be conserved. So this allows us to generate all kinds of hypotheses that we can experimentally test. And we've shown so far that there are things that are really conserved between cell cycle regulation and the regulation of sleep. The other reason why we thought cyclin A was exciting is because it's expressed in a very limited number of neurons in the brain. So we see cyclin A in about 40 to 50 neurons. Here it is protein in the cell body, and also in these puncta, which are actually clusters of dense pore vesicles, which are vesicles that contain neuropeptides that are secreted from neurons and then regulate many different processes. So here are the cyclin A neurons schematized in the brain. They're these discrete clusters expressed in this factor. And this being the fly, we have all these amazing tools that allow us genetic access to these neurons. So we can now start activating and silencing these different neural populations and then ask what happens to sleep. And we were particularly interested in these cells that are on top of the brain. They are collectively thought of as being analogous to our hypothalamus, which is part of our brain that regulates circadian rhythmicity and sleep, among other things. The part that's outlined in orange was already a known sleep center. Cyclin A is expressed there. But these two factors, really nothing was functionally, these two clusters were thought to be analogous to the hypothalamus based on development, and functionally really nothing was known. It wasn't known if these are sleep neurons. So what we did is then we decided to activate or silence these neurons, and what we showed is that when you activate just these 12 to 14 neurons, you prevent flies from sleeping, but if you silence them, you induce sleep. Okay, so manipulation of a very small number of neurons can have a profound effect on behavior. And we then, so these are the cells that I just told you about in red here. And we found that another set of wake-promoting neurons projects close to these cyclin A neurons. And our model is that during the day, these neurons in green are signaling to the neurons in red, perhaps keeping them active and keeping the animal awake. And during the night, there's actually less signaling, and cyclin A neurons are less active, these dorsal cyclin A neurons, and the animal can sleep. So this is the model that we can test. I don't really have time to go into much detail, but I just want to tell you that there's very few neurons here that we're talking about, and very complex behavior that resembles our sleep. So we really can get a good handle on how neural circuits function. And also, I told you that we see cyclin A in these puncta, which we think are secreted. And I should tell you that there's very little that's known about how these vesicles containing neuropeptides are targeted for secretion, where they need to go, because they can actually be secreted from pretty much anywhere in the neuron. So how is it known where they need to go? How much is released? And so we're thinking about cyclin A potentially regulating this, and we like neuropeptides as regulators of behavior because they can be secreted over long periods of time, so you can think of it as a sort of slow release pill, right? So sleep is a behavior that can last for many hours. So this is kind of just a little bit of a flavor of what we're doing. And I started off with this poetic quote from Shakespeare, and I want to give you something different now, which is this quote from Nabokov that says, "Sleep is the most moronic fraternity in the world. Mental torture I find debasing. I simply cannot get used to the nightly betrayal of reason, humanity, genius." What he's talking about, of course, is the fact that every night when you fall asleep, you lose your consciousness. you lose your awareness of both the external world and of your internal states. This is a profound change that happens to all of us every night. And so we want to use this dichotomy between waking and sleep to try to understand how sensory awareness arises in the brain. So essentially, we're asking how sensory information is gated during sleep. How is it prevented from arousing your brain to full awareness? So our idea is to try to wake flies up from sleep. Try to find flies that are much easier, much harder to wake up from sleep than normal by probing their different sensory modalities, say using mechanical stimulation, optical stimulation, thermal stimulation. And what I'm going to tell you about is a project mainly of my fantastic postdoc, Iris Titos. So she initiated this, and she's now joined by an excellent graduate student in my lab, Michelle Frank. And they're going to be working together to tackle sort of this different aspects of this problem. And they were helped a lot by Benjamin Sanchez, who is a postdoc at Beth Israel, and then Ofer Mazur and Pavel Gorlik. I really want to give a shout out to these guys too, who are the Harvard Instrumentation Corps and who are, making our dreams come true. They're building like any piece of equipment they can build for us that we want. So this is how we probe flies' arousal threshold, or how we try to wake them up from sleep with mechanical stimulation. So we're using these very large bass loudspeakers, and then we custom built low friction platforms to fit on top. And here on top of that are the monitors, locomotor monitors that contain tube with flies, and then we wrote a program that allows us to specify the frequency, amplitude, duration, basically the intensity of the signal that's produced by the loudspeaker, and you can have any permutation of these. So this can cause these platforms to vibrate just a little bit or a lot, to really shake, and this is what it looks like in real life. These are the loudspeakers, here are the monitors with flies, and we built many of these, so this is very automated, high throughput, and precise. So what I'm going to show you is that the system works. It works really well, and we can actually pick flies up. We can pick up these differences in arousal threshold. So the majority of animals you would look at would fall somewhere along this line. Here we have the vibration stimulus, and here we have the percentage of flies that wake up on the y-axis. So you can see that these guys that are in orange are harder to wake up from sleep. And the guys that are in blue are much easier to wake up. So this very low intensity stimulus that wakes up almost no control flies, wakes up 40% of these animals. And whenever we get something like this that's hard to wake up, we look at how these animals respond during wakefulness, because we want to know that this is not just a sensory defect. So we want to know that they have a normal startle response. Okay, but somehow their gait during sleep is different. And what is interesting is that flies can have different arousal thresholds despite having basically identical sleep amounts. Because for us, the question was, are we just reinventing the wheel? Should we just go to the known sleep factors and then look at them? And is there any predictable correlation between sleep amount and sleep intensity? But what we find is that we can really uncouple sleep depth from sleep duration. And sleep depth is really not routinely measured. So the factors that we're picking up are not necessarily what you would find if you were looking for sleep factors. Another important question for us is there a central gate in the brain that gates sensory information during sleep? Or are there peripheral gates? Are different sensory modalities gated in unique ways? So to answer that, we're probing flies with different kinds of stimulation. So as I said, we have a system for optical stimulation, and what we showed is that these guys in orange are hard, and the guys in blue are easy to wake up with both mechanical stimulation and with light, which points to existence of a central gate in the brain. So it's still possible that there is going to be some peripheral gating, but there is likely to be a central gate in the brain. And as I said, we want to build another system for thermal stimulation as well. So our idea is now to look for genes and to look for neural circuits that regulate gating. So we're basically eliminating functions of various genes, and we're also activating and silencing different neural circuits and looking for those that are involved in gating. So this is a non-biased approach, but I'll say that we're also studying one factor in an unbiased way, or we picked one factor to study, which is dopamine. So what was known, both in the fly and in mammals, that if you have a little bit elevated dopamine at the synapse, your arousal threshold is lowered a lot. Meaning that a low-intensity stimulus comes in, and now your brain interprets it as if it was a high-intensity stimulus. So how does this happen? And again, working in a fly that has about 200 dopaminergic neurons, we have access to most of these cells. And we can actually now ask which the neurons are that are important for gating. So this is what we're doing now. We're trying to figure out exactly which of these neurons are releasing dopamine that is involved in sensory gating and what the downstream targets are. And this is, I'll say that this is very important for human health. So dopamine is a factor that's involved in arousal, both arousal from sleep and also different levels of arousal during wakefulness. So there's a hypothesis that people with ADHD are actually hyperactive because they have abnormally low dopamine tone, abnormally high arousal threshold, so they're constantly self-stimulating in order to get over the threshold. So we really want to understand how this works, and I think we'll be able to dig deep. And one last thing that I want to tell you is that what we realized when we have flies on these platforms that can vibrate, we can use this to look for homeostatic factors. So if we deprive flies of sleep, either overnight or in the second part of the night, you see that they sleep now during the time that they're normally active, right? So they sleep here. So what if we, after probing the arousal threshold with sporadic stimulation for several nights, we just crank up the stimulus, we prevent flies from sleeping, they're already on these platforms that can vibrate, and then look for animals that are having trouble recovering from sleep loss. Because we all know what happens when you don't sleep. We all know that groggy, awful feeling that follows a night of inadequate sleep. But what is that feeling? Where does it come from? And how do we deal with that? How do we know what to do with that? Okay, so the idea is, again, to look for homeostatic factors. And here's an example of what a homeostatic factor would look like. So these guys, after sleep depriving them, they just continue walking around. Okay, compared to the controls which sleep, these guys just continue walking around. So they can't tell that they've been deprived. They don't care, or they don't know what to do with this, right? So we want to find these kinds of factors, and I should say that there are very few true homeostatic factors that are known. Things that wouldn't necessarily be affecting baseline levels of sleep, okay? Because baseline sleep is normal in these animals, but it's this deprivation that they don't know how to deal with. And again, keeping in mind that sleep is very, very, very similar in flies and in us, we really hope that the system is going to help us understand sleep on a deep molecular, mechanistic, and also circuit level. And with that, I want to thank the people in my lab. So I didn't have time to tell you about most of these people. So Hannah will be working on the Cyclin A project, and Iris and Michelle on the arousal threshold. And then I, again, want to thank Benjamin, Ofer, and Pavel, who have helped us a lot with building the equipment and that we need for this project. Thank you so much.
Can you explain again how you actually measure sleep?
How do you measure sleep? So we routinely measure locomotor activity, and this is because it's been shown that there's an excellent correlation between locomotor activity and electrical activity. So you don't really need to. Flies are small, and I would say this is the main disadvantage of the flies, that it's hard to do electrophysiology routinely the way that you can do in the mammals, right? Just put things on the scalp. But since there is such an excellent correlation between activity and sleep, we can just use it as a proxy for it. And then if we want to look at things in more detail, you can measure electrical activity.
Yes. My training as an obstetrician and gynecologist makes me ask this question.
Okay.
When you're a young mother, before you have the baby, you can sleep very well. The minute you have the baby and it cries, you wake up. So it's a change in what you call, the threshold. The thing that fascinates me is if somebody else's baby cries, you don't hear it.
Absolutely.
And I've always been sort of fascinated. Now that's different from some other neurologic responses, which is a letdown, which if a cat cries and sounds similar, you let down whether it's a cat or your baby.
Mm-hmm.
So I'm kind of puzzled as to the discrimination in that threshold.
Yeah.
Now I know your flies don't.
No, but they may. So this is what you're talking about is there's the intensity of stimulus and there's also salience of the stimulus, right? And this is definitely something that, that we're interested in understanding, how it is that, exactly, if somebody whispers something to you when you're asleep, you don't react. But if they whisper your name, there's a higher chance that you will react, right? And if your baby cries, like you said, there's a higher chance. And we really don't know how this works. So we think of thalamus in our brain as being a gate, but it's not a perfect gate. It's not an on-off switch. It really, you know, lets things through still in a context-dependent manner, and it's really not known how this occurs. So we've established the system in the fly exactly to ask these kinds of questions. So for instance, you know, you can train flies with a stimulus that would be pertinent to them, and then try to, you know, apply that stimulus during sleep and see if they would respond. Maybe if you change the context, you would kind of change the response and then figure out where that's gated, where it goes through. So I don't know how it's done, but hopefully we'll find out.
That was an excellent talk. I have
Thank
two
you.
questions for you. One, I realized that there's a term for what I suffer from each night, which is sleep initiation. So I wonder if, you know, you gave your flies coffee and things like this. I wonder if you've tried exercising your flies to help them with their sleep initiation problems, and if you've seen
I
any.
have not tried that. No, I have not tried that. I have to say they're exercising a lot, actually. When they're in those tubes, they're walking around a lot. But yeah, I'm glad you say that. So you have trouble falling asleep. I have trouble staying asleep. And that's great. I mean, many of us in this room probably have problems with one or the other, right? Some people have both, but it's usually one or the other. And I'm just hopeful that, you know, by doing genetics and looking at circuits, we'll be able to understand what goes on.
So obviously some mornings are greeted with me being sleep deprived, but I know by the afternoon I will always feel okay.
Yes.
So is there anything, if you can keep your flies up, do
Right,
some of
that would be
them,
done.
can you detect any change that indicates that maybe they're happier being awake?
So, you know, I would bet that, well, you could give them some, for instance, learning or memory tests, you know, or like odor discrimination or whatever, see how we have this thing we call the fly Olympics, or, you know, so you can put them through all kinds of tests and you could measure exactly how they perform, you know, after deprivation in the morning. And I know what you're talking about, but I bet that still, if you look at their, you know, lifespan, it would be, regardless of if you feel okay or not, if you were able to get over that groggy and awful feeling, your health would still suffer. So there would definitely be consequences to sleep deprivation.
I'll keep that in mind.
Yes, absolutely. There's an effect in the flies on lifespan. Yes, deprivation.
A very nice talk. I am a born mentor. I can't help myself. So I love this statement about the homeostatic factors. And you say that there are so few homeostatic factors. And I like to suggest, both for all the scientists and you as well, I unfortunately didn't organize myself to show you the data because it's a bit complicated. But in cancer, what we find is that everything relates to everything else in relation to structure. Mm-hmm. So when people, if people did the experiments in a fly or in 3D rather than a flat dish, you find that all the signaling pathways are connected. So when we revert the malignant phenotype, we use this agent, everything else goes down. We do this one, everything else goes down. So to look for a single factor is not going to work. So the thing to try and see, I mean, it is important, but what would be great is that if you could look at the entire metabolic or gene expression or whatever as you find these different symptoms, because homeostasis then is a measure of all those different factors that keep the balance. So I would suggest, and we find this again with cancer, that what really matters is the balance. Mm-hmm. In other words, we can take something that is completely off the back, but then bring other things up, the cell thinks it's normal. So it's the ratios of one thing to the other.
Yeah.
So I like to talk about this sometime later, but I don't. Yeah. I know we are running late. So
well, I don't know, do I have time to answer? Well, of course, I think that there's going to be many, many things that are affected. And we look at this just kind of as a gateway for us to get a handle on the problem. And of course, you know, we don't think it's just one thing. I mean, there's no such thing as just one gene. But we know, as I said, that there are genetic sleep disorders that tend to run in families and that are due to mutations of single genes, which then of course affect many hundreds or thousands of other things. But you got to start somewhere. So we like to look at pathways. So for instance, you know, we study cyclin A, and we know all the different things that are interacting with it in cell cycle regulation. So we go and actually look at the entire networks of things to try to figure out how things work. So of course, it's many, many different things.
Thank you very much.